Hello children, we are watching Vivid Botany. Under this we have a question today about the citric acid cycle. It is very much important question for competitive as well as for any kind of exam of the +1 and +2. It comes under plant physiology. Now let's have a look at the citric acid cycle. It's also called as Krebs cycle because it is discovered by Sir Hans Krebs. It's also called TCA cycle, that is tricarboxylic acid cycle. Tricarboxylic acid cycle, or it is also called TCA cycle. Now, in this, let us see what happens exactly one after the other. First, it is the oxaloacetic acid, which is a 4-carbon organic acid that combines with water molecule plus acetyl CoA, which is 2-carbon organic acid. In the presence of the enzyme citric synthetase gives rise to citric acid, So since of course coenzyme A is liberated out, so citric acid is 6-carbon organic acid, and we know oxaloacetic acid is 4-carbon, acetyl CoA 2-carbon, total it is 6-carbon citric acid is formed. And now the citric acid undergoes dehydration. That is loss of water molecule minus H2O that I am writing, to give rise to cis-aconitic acid. So this is also 6-carbon organic acid. The enzyme is here aconitase. This is the name of the enzyme, aconitase. Now what happens? Cis-aconitic acid will convert into isocitric acid, and here that reaction is hydration, that is addition of water in the presence of enzyme, the same one, that is aconitase. So remember, citric acid is 6-carbon organic acid, cis-aconic acid is also 6-carbon organic acid, isocitric acid also 6-carbon organic acid. So remember, therefore there is no so far loss of the carbon dioxide. Now the isocitric acid undergoes the first oxidation. Remember, oxidation means loss of hydrogen. So this hydrogen is accepted by NAD plus to give rise to NADH. It is NADH. It is formed here. In fact, it is NADH plus H plus. So it is first oxidation here, and name of the enzyme is isocitric dehydrogenase. Right. Now, after undergoing oxidation, isocitric acid will convert into oxalosuccinic acid. So oxalosuccinic acid is also 6-carbon organic acid, because so far there is no decarboxylation in isocitric acid. This oxalosuccinic acid now undergoes decarboxylation in the presence of the enzyme oxalosuccinic decarboxylase. I write here DC. So 6-carbon oxalosuccinic acid now, after decarboxylation, that is loss of carbon dioxide, to give rise to a molecule of alpha ketoglutaric acid. Which is 5-carbon organic acid. Okay, so this alpha ketoglutaric acid undergoes here oxidative decarboxylation. Yeah, this is more important. It undergoes here oxidative decarboxylation. So you must be knowing here oxidative decarboxylation means it is loss of hydrogen as well as loss of carbon dioxide. That is, oxidative means loss of hydrogen. Hydrogen is lost here. This hydrogen is accepted by again NAD plus to convert to NADH plus H plus, and decarboxylation, that is loss of carbon dioxide. So it is loss of hydrogen and loss of carbon dioxide. So after losing this hydrogen and carbon dioxide, the thing that is formed is here succinyl CoA. Succinyl CoA in the sense succinyl coenzyme A. So alpha ketoglutaric acid is 5 carbon organic acid and succinyl CoA is 4 carbon compound, right? So here the enzyme is alpha ketoglutaric dehydrogenase. This is the name of the enzyme. And one more thing here is that addition of CoA is also there in this reaction, because in succinyl CoA we find CoA here, right? Now this succinyl CoA undergoes what do you call thiokinase, that is splitting, it takes place. It undergoes splitting. Splitting in the sense succinyl CoA will form here 3 compounds. That is the magic, remember here. It will form here 3 compounds. What are the 3 compounds? One, first, CoA is liberated. Coenzyme A is liberated. Next, here inorganic phosphate is added to GDP. As a result, GTP will form, that is guanosine triphosphate. But again, guanosine triphosphate will split into inorganic phosphate and GDP, and inorganic phosphate is added to here ADP. As a result, ATP will form. So this formation of ATP is called as SLP, that is substrate level phosphorylation, right? So after formation of the GTP and form liberation of CoA, the remaining thing that is formed is here succinic acid. Succinic acid, which is 4 carbon organic acid. Now this succinic acid will form into fumaric acid. It will form into fumaric acid. Here third oxidation takes place. So you see here this is where this is first oxidation, and this is second oxidation. The second oxidation this is, and here third oxidation takes place. And here oxidation means you know that is loss of hydrogen accepted by FAD to convert into FADH2. It's not FADH, it is FADH2 that is formed. The enzyme is succinic dehydrogenase. The enzyme is here succinic dehydrogenase. Now this fumaric acid, which is of 4 carbon organic acid, will convert into what do you call that malic acid. Fumaric acid will convert into malic acid by the reaction of hydration, that is addition of water molecule in the presence of enzyme fumarase. Now the 4 carbon malic acid will convert into the oxaloacetic acid. It will convert into oxaloacetic acid, and again this is here fourth oxidation. It is fourth oxidation. So we are seeing here 4 oxidations. The enzyme is malic dehydrogenase, and the last hydrogen is accepted by NAD to convert into NADH, NADH plus H plus, right. So this is like this here, 4 oxidations takes place. This is called as the so-called Krebs cycle, okay. Now I wanted to explain you the total sum of the reactions in the Krebs cycle. I think it's elaborate. You can take the screenshot of this also here. You can take screenshot of the Krebs cycle here, but that is getting minimizing. Now you can take screenshot of this, right. Now let's see the question here. The number of substrate level phosphorylations in 1 ton of citric acid cycle is, yes, this is what the core point of the question here. Now remember, phosphorylation, I will go to the bottom level of the concept. Phosphorylation means addition of inorganic phosphate, is called as phosphorylation. Here, in terms of the metabolic reactions like photosynthesis and respiration, the addition of inorganic phosphate is to ADP, that is adenosine diphosphate, to give rise to adenosine triphosphate, which is called as cell currency, or it is called as mobile currency of the cell. So this phosphorylation takes place in 3 types. Let us see what they are. One is photophosphorylation. What is new here? Photo is new. So here this is addition of inorganic phosphate to ADP to give rise to ATP in the presence of sunlight. That is, formation of ATP is driven by sunlight. That is, addition of inorganic phosphate is driven by sunlight. That is H nu. It is called photophosphorylation. This takes place in chloroplast in the presence of sunlight as part of photosynthesis that takes place in green parts of the plant. Second one is oxidative phosphorylation. Oxidative phosphorylation means it is also addition of the inorganic phosphate to adenosine diphosphate to convert into adenosine triphosphate. So this is driven by oxygen. Okay, it is oxygen driven process. It is driven by oxygen and this is driven by sunlight. So oxidative phosphorylation, that is driven by oxygen, takes place in mitochondria. As part of the respiration, right. This is one thing. And next, third one, that is substrate level phosphorylation. This is what the question is being framed on this. So in substrate level phosphorylation, what happens here is that when one substrate is converted to other substrate, see for example, this is substrate 1. So substrate 1 is converted to substrate 2. One substrate converted to another substrate like substrate 2. During this, inorganic phosphate is added to ADP to give rise to ATP. This is called as substrate level phosphorylation, or simply we call it as SLP. So where does it takes place basically? If you see in glycolysis, it takes place in glycolysis, that is during the formation of 1,3 bis PGA to 3 PGA, 1 comma 3 bis PGA to 3 PGA. Another is phosphoenol pyruvate to pyruvate. During the two reactions it takes place. Another example that I give you in Krebs cycle. So this is what the question is framed here. Now you see in Krebs cycle we are looking here that here where this I wrote here, you see. Let me take there. When succinyl-CoA converts into succinic acid, the first formed cell currency is GTP. Then from GTP again the GDP and IP both are separated. This IP combines with ADP to form the ATP. This is called as substrate level phosphorylation that takes place during the Krebs cycle, that is citric acid cycle. Yes, so I write the same here. That is here what happens during Krebs cycle during cleavage of succinyl-CoA. During cleavage of succinyl-CoA, the so-called ATP here is formed, which is called substrate level phosphorylation. So I think you have understood here. He is asking the number of substrate level phosphorylations in 1 ton of citric acid cycle. So we are looking here the 1 ton of citric acid cycle. In 1 ton of citric acid where there is only 1 ATP is formed here in this reaction. So the answer is absolutely second option, that is the answer. So this is one kind of question that is being asked on metabolism, that is citric acid cycle. Little bit I have elaborated. If you remember it, it's okay, but even then I suggest you to remember it total, because when they go to deeper levels, I mean depth of the concept, you are supposed to be ready to answer the question. Okay, so hope you have understood the Krebs cycle, that is citric acid cycle. If you do not understand, once again I am appealing to drop a line, to make a comment and give your response in the comment box of my YouTube channel, that is Vivid Botany. Okay, so thank you very much, and I appreciate your preparation. I appreciate your watching of my channel, YouTube channel, and I appreciate if you really ask any doubt regarding any concept of botany, which I am ready to explain the nook and corner of the subject. Okay, so do subscribe my channel Vivid Botany. Thank you.